Christian Forst is assistant professor at the Genetics and Genomics Department here at Mount Sinai. Christian, even though he's an assistant professor, he has a very extensive career and a very distinguished career. He received his education at the University of Vienna, Austria. He received a bachelor's degree in astronomy and then a master's degree in chemistry and a PhD in theoretical chemistry. From 97 to 99, before he joined the Beckham Institute in the University of Illinois, he was a group leader in the Institute of Molecular Dynamics in Jena, Germany. And then he came to the United States to the Beckman Institute in the University of Illinois. And then 2 years later, he moved to Los Alamos National Laboratory, where he spent 10 years there. He was faculty for 4 years at UT Western. And in 2012, he joined Mount Sinai, where he's doing bioinformatics analysis, and he's interested in studying influenza and also using transcriptomics data. And today he will tell us about his work and also how potentially his work is related to the LINKS project.
All right, thank you, Avi, for this kind introduction. Yes, I'm working and still continue to work on influenza as well as on other infectious disease projects, but mainly, as you already see in the background, I am interested in influenza, the influenza infection and the processes that's required by influenza to infect the host, but also, of course, the host defense processes that the host tries to throw against influenza to prevent infection. The particular talk I would like to present today is now in press in one of these major partner journals, and hopefully it will be published pretty soon. Let's see. Oh, it takes a while. All right, so I would guess the auditorium is quite diverse. I just give a brief introduction. Influenza itself is a negative strain segmented, negative sense RNA virus. It consists and has a segmented genome of 8 segments and about 11 proteins, depending on the particular strain. It can be 12 proteins, but mostly 11. The two main proteins, the surface proteins that actually give more or less the strain names, the H and the N, you may already recognize. These are the hemagglutinin for the H and the neuraminidase for the N. And of course, they are serologically important, and the host immune system responds to this specific surface proteins, and that's why there's this classification of H1N1 or H5N1 of the different flu strains. But there are also, of course, other proteins that are required and used by the influenza particle to hijack and modify the host processes. They are important in the different phases of the life cycle, and I have another slide for that. In particular important is this non-structural protein, which actually is more or less, I think, the key protein of the virulence of influenza, because it modifies a lot of key processes, in particular the splicing and splicing machinery to benefit the viral mRNA processing, but it also has many other functions. It's a multifunctional protein. All right, so far, excuse me. So influenza itself has a wide host range, of course, all vertebrates, but mainly mammals and birds. The native host seems to be the wild waterfowl, so mostly duck, but of course it can infect many different other organisms, and they of course depends on the specific serological footprint of the different virus strains. So there are endemic virus strains that can of course infect hosts within a particular organisms and species, but of course zone exchange that can jump hosts, and you may already know H5N1 is, for example, the flu, the bird flu, which can of course jump between birds and humans, but on the other hand may not be able to, a bird infected human may not be able to infect another bird flu human, but nevertheless it really depends on the specific strain. What's particularly dangerous, and at least where there's large concern of re-emerging new highly virulent flu viruses can emerge, is, for example, a swine, which actually can reinfect humans in both of swine virus, or infected swine can infect humans and vice versa. In particular, if, for example, pigs are infected by multiple different influenza strains due to this segmented genome, there can be a rearrangement, and actually through this rearrangement from different influenza virus strains, new viruses can emerge. And so maybe, for example, this happened during the swine flu pandemic that through multiple infections in pigs, this new virus emerged and could then overcome the human immune response. All right, so sorry. The main focus on this particular study is just cell-based. So we're only looking at the infection of a particular host cell or host cells, and in this sense only look at the innate immune response of this host cell. So we are not interested, or we disregard any other higher order adaptive immune response. So we only look at the response and the machinery within this particular host cell, and usually the particular host cell for the influenza infection. The first sites are lung epithelial or bronchial epithelial cell. So in this sense, we are looking and focusing our studies on these particular cells. Oh, maybe I just okay. So, and what you will see is, so this is just the cartoon of a life cycle. This is taken from the review by Watanabe in 2010. You see that a viral can attach to the host cell, and by using all these different host factors, and the color code is just, there are known host factors in this at 2010. There are of course many more host factors known now, but nevertheless the yellow ones were the known host factors at this time, and the red nodes indicate the host factors that were identified by this large scale high throughput experiments, mostly done by siRNA screens that were reviewed in this particular paper. And so there's a entry phase, a transport phase, so the virus particle get then and the RNA out of this virus particle get extracted, get transferred to the cytosol, to the nucleus and then import into the nucleus by using these nuclear pore complexes. In the nucleus, of course, there is a machinery to transcribe the viral RNA, to copy the viral RNA, but also not only copy them for further usage for a duplicated genome, but nevertheless also then to further, of course, translate the particular viral proteins. And so there are again other host proteins, in particular the spliceosome and all the proteins involved in splicing and mRNA processing. Again, this processed mRNA is then exported out of the nuclear pore complex, as I said, translated the proteins that are required for as viral proteins for the newly assembled viral vesicle, but also, of course, the RNAs, the viral RNAs are transported, they are reverse transported out of the using the Golgi complex, and then a newly viral particle bulges out of the host cell, and of course not just one but many. And all these processes are actually modulated, of course, by the viral proteins, and of course then there are additional processes by the host that trying to get rid and prevent these viral processes. So it's a fight between two systems: the viral particle and the virus, as well as against the human host. So what we are doing here is, so we have a pipeline or a tool of methods and a particular approach to identify and analyze these large data sets, and of course to use this approach to identify key processes, key genes, key targets that can explain and actually predict new targets in this particular scenario. In our sense, of course, in this scenario is targets that are required or are interested during influenza infection on both sides, interested for the virus for survival and replication, but also then interested for the human host for defense. And in this study, we're looking at and using publicly available transcriptomic data, as we already mentioned. That this is one of the main source of information, at least one main source of information what we're using here in this study. We used different sets of influenza infection data sets, transcriptome profiles, and not only just one, but many, because we were interested in the consensus response. We want to look at the commonalities between different scenarios, in particular between different flu strains, but also slightly different, not really tissues, but slightly different cells as first line of location where infection occurs. And so what we're doing is we have a overall consensus, excuse me, consensus analysis where we identified the differentiable genes. So we're looking at consensus gene response, and because we have not only just a simple response measured in our data set, but all these data sets are time series measurements. We were actually interested in the time series response. So what we have and what I call here significant response genes, SRGs, are actually the responding genes that are significantly responding through a time series. So this could be whatever, a upregulation and then a downregulation afterwards. So it can be just a response, a change of expression levels during time. We also want to be more specific or slightly differently, where we're looking at the trend, up or down regulation. So you see, it's just a cartoon and very small. But nevertheless, we are also very interested in an upregulation, in a significant upregulation across time series or downregulation across time series. This is then, of course, looking at slightly different genes, because significantly responding genes can, as I mentioned, up and down regulate, so they may not have any trend that's only up or only down. This consensus approach also we expand to networks. So we looking at and constructing, inferring co-expression networks, again, not just one, but for all our data sets. We try to relate them and rank these sets, particularly not only the sets, but specifically co-expression modules, and I have some more slides on that, because it's not just simple modules, but again consensus modules in these networks. We then have and can identify by this ranking and by further topology estimations and analysis particular key regulators, where we focus on a particular key regulator and use this as entry point in our analysis, and all the within this network neighborhoods, all the genes that are influenced by this key regulator. And of course we want to do a validation. So what we then did is this particular key regulator will be knocked out, and we use CRISPR-Cas9 for knocking out this particular gene, and then checking the response both on the some physiological level, if in the case that how this knockout influences the influenza life cycle, but also of course the downstream knockout and the response of the genes. And this is some of the overview what this study is some based on. So these 12 data sets are just from GEO. They have is to some extent a mix and match between different virus strains and cell lines. So in particular, there are different flavors of H1N1 viruses, and on the right side you have the table that shows this different virus strains, but in particular not only H1N1, but we also have H5N1, H5N2, and H9N2, which are all these bird flu strains. We use 3 different cells. They are 2 cell lines, A549 and KLO3. So one, the A549 is a lung cancer cell, the other KLO3 is a bronchial cell, but we also have primary cells. This is the human bronchial epithelial cell. And then we also look at different levels of infection. This is the multiplicity of infection, this MOI, and in our case ranging from 0.1 5 is already rather large and high, but nevertheless for this HPEG it was apparently necessary. But nevertheless it could be, well, it's not problematic, but we have this range of different MOIs, so we, and with our consensus approach, we can cover all these different MOIs. It's to some extent problematic because, of course, if the MOI is too large, meaning if the virus overwhelms the cells, then the cells could just start starting in apoptosis and shuts down the whole cell machinery. And again, that's what I mentioned. We are using time series data. So just to give some feeling of how similar, different these responses are. So this heat map just shows the significantly responding genes of all these 12 data sets, and the color codes on the edges. So on the top and the left side just indicate the different scenarios. The top are just the different cells and cell lines, and on the left side, they just show the different viral strains. But the take-home message here, and this is just the overlap based on Fisher exact tests. So the greater the more significant, and the small numbers are just the fraction of the overlap, so the fraction of intersection based on the whole individual 2 sets where they are checked. And so the take-home message is that they are, to some extent, true, there are some differences, but mostly similar, and the similarity is more based on the cell line and not so much on the viral strain. So, for example, the KLO3 cells have both H1N1 and H5N2, but they mostly, at least the overlap is mostly significant. Sure, the overlap size is a little bit smaller, but nevertheless, there's some significant overlap. Same thing for A549, they all have A549, again, H5N2 and 1N1, which of course goes up here. This is the H1N1, there are 2 for H549. So in this sense, they are rather similar. The outliers more HPEG, which is somewhat much different. It doesn't have really any overlap with the other one, but again, explained that apparently the cell line is dominant in the response, not so much the different viral strains. So the response itself and the significantly responding genes then can be identified just by the number of experiments and data set where they're responding to, and it's just ordered by this consensus and the number of the consensus sets they are occurring. And then the highest number is this heterogeneous nucleoprotein. So this is a component of the spliceosome in the nucleus, and it shows up. So it's significantly expressed in all 12 data sets. So this is based on the SRGs. This up and down refers to this trend analysis, and this is just to show the fraction of data sets where it's actually up and down regulated, so meaning in 75% of the data set is down regulated and only to 17% up regulated. The next one, so all others are significantly responding in all but one data set, so including, for example, this low GTPase activating protein, but there are also chemokine CCL5, which is responding some of them are mostly downregulated. This one is actually exclusively upregulated in all data sets. So these are some of the different information shown. So these are to some extent the main common responders during influenza infection. So of course one can similarly identify the whole gene set, not just individual genes, and looking on gene ontology, functional assignments. Again, this can be done based on all these different consensus levels. So this is just this curve on the right just shows the number of significantly responding genes based on the consensus. In the course of this study, we actually then focused just on the consensus 7. There are one more than at least half of the data sets should respond. So this Venn diagram also focusing on the consensus 7, and what you would expect is that the most common response, for example, on significant responding genes are this interferon signaling, and of course not only just responding by itself, but they actually both significantly responding as well as upregulated. So in the intersection between the upregulated and upward trending genes and the significant trending genes are this type 1 interferon signaling in response to virus. But of course there's some other functions, so downregulated are mostly these lipid metabolic processes or localization. So the localization of proteins and cellular components are shut down in this analysis. And of course this is just a very crude picture, but it gives an overall picture what's going on, to some extent reassuring what's already known. But on this high level one can identify what biological processes are either activated or inhibited during influenza infection. So the next level, and again looking at consensus analysis, is using co-expression networks, so similar to the identifying consensus differentially expressed gene. We were looking at consensus co-expression networks, so we're using WGCN A as inference method. We tried out some other methods, but to some extent co-expression and correlation work best in our case, which was rather interesting. But we used this WGCN A, again built these 12 co-expression networks, and what this small color bars on the side indicate, and we already know when you know WGCN A, so we use not only the network but actually can identify co-expression modules, so small clusters of genes that are expressed together and assumed to have similar function. And again, we're not focusing on individual networks or individual modules from these studies, but actually look at the consensus. So we, similar to this clustering of co-expression genes in modules, we actually cluster these modules in consensus modules using a different metric and another metric for this clustering, and ending up in this consensus modules again from 1 to 12 as consensus variable, having common gene modules from 1 to in all 12 data sets. And again, you see they all coming from different strains in our different cell lines. So these modules then of course can be again identified for functionality. As I said, these modules, these consensus modules may show and tell something about what functions are some are triggered by this infection process. They can be ranked, and they are just ranked. So they're ranked by response and overlap between the modules and the differentially expressed genes. So just the idea behind this, that the more significant or the more differentially expressed gene sets are intersect with the particular modules, the more significant and higher rank this particular module is, and it's just shown here in this circus plot. So you start with the most significant and highly ranked module up on the top, and then clockwise the rank goes down, which is also indicated by this black outer line here. So the larger this bar, the higher the rank, and all these other heat maps indicate the overlap between the modules, so the consensus modules in the particular DG. So you see the ranks up here, the particular nomenclature down here. So I have individual SRG enrichment from the different data sets, a consensus DG as well, the up and down one trends. And on the inner ranks, I just check, and inner rings, I just check about the enrichment of known signatures of influenza infection. And so all the best modules, or the highest ranked modules, of course, are all significantly enriched, but the Gs as well as have something to do with influenza infection. That's what you, to some extent, expect. And the functions are just shown here, so from the highest rank to lower rank, and this is just what I refer to best modules or consensus modules shown here, based on, again, viral reproduction, influenza life cycle, which is mostly the ribosome, but there are also cell cycles. This is the Aurora A pathway, which is some mitosis-based mitotic spindle process, but there's some other processes, so some signaling MAP kinase pathway. What's some interesting is also this Golgi-associated vesicle biogenesis is important. So this is this vesicle transport in the cell. And so this gives another picture on another level, and particularly on the network level, what's going on. But we want to continue. We want to identify particular key target, key driver, and induced processes. So what we did is we actually looked at the connectivity of all the targets in these networks, and again we look at the consensus. So we look at the cumulative connectivity of genes in all these networks, and then actually combine them with our responding genes. So what you see here, the genes are ranked based on the connectivity, but they are also highly responding in most of the data sets. And what shows up, okay, MDM2 is the highest rank based on the consensus or cumulative connectivity, which is this E3 ubiquitin protein ligase. But we decided not to study MDM, particularly because the many other studies already done. A novel gene which was not related, there were no real publications and studies going on or found with respect to influenza is DOG5, and so we focused on DOG5 and looked at how DOG5 is involved in the influenza life cycle. And so all the studies now working on DOG5 assumption and hypothesis that DOG5 is a host factor and induces many other and modulates other host factors in this influenza life cycle. So how to do that? And in this place, we want to use our network analysis and network topology and combine them with perturbation data. And so we have our consensus network, and you see this animation. So this should be, for example, DOG5, but can be any target that has to be tested. And so we look at the network neighborhood of a particular target gene and check for each cumulative network neighborhood if this network neighborhood is intersected and overlaps with a particular signature genes. And in this sense, for example, links is very useful because links uses, of course, as you well know, perturbation analysis. So using the particular a particular gene in the network with the corresponding gene that is knocked out in the links data. That's some of the idea behind our analysis. And it works somewhat pretty nicely, but this is some of the idea to identify and assume and predict the potential effect of the target on the overlap genes in the neighboring network. So what we did, induced links data set. As I said, they're rather useful. They have to be matched, or at least we tried to match them, of course, with our gene expression data set. So we found at that time when we did the analysis, found this two matching links data, so E549, which is the data set that we're already using by part in our expression data from GEO, but we also have a HCC55, which is also a similar non-small cell lung cancer-based cell line. On the other hand, I also tried out a breast cancer cell, which of course has nothing to do with influenza, but nevertheless it has similar number of genes involved, and was just curious what's going on. And the interesting thing is that we actually found some genes. But nevertheless, what you see here is this curve is just the enrichment and the minus log p-value of the best responding gene in the network neighborhood. So again, what I shown in the previous slide is we focus, so we use actually all the genes that are available, meaning the intersection of the genes tested by the links data and the genes that we have in our network. So in this case, about 2,500 genes. And for each gene, we check if the particular gene in the network and the neighborhood has a significant overlap with the responding genes from the links data. And we look, of course, at response both on up and down regulation. So if the genes in the links data after knockout are either upregulated or downregulated. And so we do this for the network layer, of course, because we have a finite network. At one point, we actually saturate our network, so that's what would happen. For example, I think it's step 6, otherwise we won't get any significant response anymore. But nevertheless, what you see, so in network 2 is some of the peak of enrichment and in these different networks. So the best responding data set would be the A549 knockout links data sets, and the best overall response is this proteasome subunit, which is actually required for influenza infection. Now I'm coming back to my breast cancer cells, because it's interesting in these breast cancer cells I have this ATP. This is a V ATPase, which is actually important for the influenza import. Apparently, it's obviously it's just this V ATPase is important for changing the milieu in the endosome. So this is apparently rather universal on different cells. That's why it also shows up in the breast cancer, and apparently it shows up better in the breast cancer cells than in the other cells. But nevertheless, it was somewhat peculiar. The one thing that is useful the links data is, the links data, of course, having the combinatorial complexity of having all different types, not only all the genes knocked out, but also other different conditions. It cannot cope with all the specific scenarios, what, for example, we are looking at, and the one particular scenario is, of course, infection. So the links data is at least for now, and I would guess in the near future, baseline, mostly baseline, and so it is useful, but for further downstream analysis, one then needs to require some other more targeted approaches, and that's what we then did. So in similar sense of the links approach, but only for one particular target, and in this case our top 5 target, we were looking in using a knockout, and using the knockout in a 549 cells orchestrated by the CRISPR approach, and the knockout is done in A55 cells using two influenza strains. So we have now two variables. The one variable is wild type versus knockout, and the other variable is infected. Infected versus non-infected should be there. And so what you see here in the curve, these are the viral loads measured by this DCID 50 screens, and this just shows some of the physiological or cell physiological phenotypic response of the cell, and how we read it. So we measured 3 days, so day 0, which is non-infection, and up 1 to 3 days post-infection. We use different clones, CRISPR clones, and compared them to the wild type. The scale on this on the y-axis is actually log 10, and so what you see here is for H1N1, the one particular clone we actually picked out is 2.64 change between wild type, which is the black line, and the orange line, but there are some better clones, worse clones. We actually used pick DOC5 C28 because it's overall the best for both virus strains. It's a little bit worse for the 8.3 and 2 strain for 1.72 fold reductions, but nevertheless, and that's log 10 value, but nevertheless it was acceptable to use for further analysis. This figure shows shows the intersection of multiple sets. It's based on the super exact test by Ming-Hui Wang, and this was published some time ago. But nevertheless, you can, so the horizontal line shows and it indicates the individual gene set, and all these vertical dots. So whenever there's a green dot, I show the intersection, in this case, between the differentially expressed genes of the H1N1 down regulated genes between wild type and knockout, and the H3 and 2 down-regulated genes between wild type and knockout. So meaning that the genes are more expressed in wild type and less expressed in the knockout scenario, which are indicators down. And again, up and up is conserved. Interesting also to some extent the mock down with H1N1. But these all are the gene sets which are significantly enriched, and where the overlap between these gene sets are significant after a tissue exact test. And down here the other actually the best functions based on the of the overlap. So again, transcription, but also sequence development process, but mostly transcriptional functions are indicated and influenced by this knockout. All right, so what just to repeat the analysis. So just the genes that are influenced, the overall genes influenced by this DOC5 knockout, without in the baseline, so without infection, it's cell migration, cell receptor signaling, communication, and extracellular matrix receptor interaction. Inducing and including influenza, it's to some extent rather similar, but it's mostly cell adhesion. So that in interbeam pathways that are induced by DOC5, meaning they are upregulated or a higher expression in the wild type with DOC5, and less activation and transcription in the knockout scenario. The opposite is the suppressed one, and there are aminoglycan processing, lysosome functions, phospholipid metabolism. So this is just a function of the overall DOC5 knockout effect. But we're also interested, of course, the combined effect and the overlay between the DOC5 network and the co-expression network and this particular knockout genes. And so what we did is we have our centered network enrichment, and again, similar to the using the links data, we used the now our own knockout data and combined them and overlaid them with our network. And this is just a slightly different view of this enrichment. Here we have, on the x-axis, the consensus set, again focusing on consensus 7, but nevertheless we showed some other consensus. And again showing the enrichment p-values as log 10, minus log 10 p-value between the our sets. And here I only look at the two sets, the knockout during the DOC5 knockout during H1N1 infection and DOC5 knockout during H3 and 2 infection. So always compare wild type versus knockout, but the additional conditions H1N1 infection, H3 and 2 infection. And when you remember the data sets, so actually I have many more. So I have about, let me go back. Here I have about up to minus 80, minus 90 enrichment, but with the particular additional conditions with infection, I can go up to that it's 300 because it's just the consensus, but here I have about 100, so a little bit better. Let me be, yeah, order magnitude better compared to the other data set without the additional conditions of infection of influenza virus. So we're using this type of data sets and then want to understand what the particular functions are. Again, just the overall functions, and now we're looking at the overlap DOC5 network and the corresponding DEGs, in particular the knockout DEGs. And again, using the super exact test, we're looking at the specific significant overlaps. This is the line here is the network, and the line up here is the DOC5 wild type and knockout DEGs, and we see quite some significant overlap. But in addition to this knockout DEGs, we also look at, of course, our DEGs measured from the transcriptomic data, and again there's some correlation and similarities of this particular genes between the network, the knockout, and for example some particular trend. This can then make some sense and identify the particular functionality of these data sets. So again, this is slightly different, only looking at the network and the transcriptomic based DEGs, but nevertheless what you see in the network is the overall response again is interferon signaling. Of course, the upregulated genes in the network are also interferon signaling. The upregulated genes, the downregulated genes in the network are membrane organization, which to some extent also is important for this DOC5 response. But DOC5 is has something to do with membrane and active cytoskeleton. And then there's many other functions that we already showed before, interferon signaling. So what we again found, so we want to look at the particular functions, not only on this overall way, but actually the specific functions that are influenced by DOC5. And I repeat myself, so I use this consensus 7 network, which is centered by DOC5. I have my additional signatures of these knockouts data sets. I can have the knockouts of non-infected, infected. We'll look at the infected scenario, both H1N1 and H3 and 2, and then look at the up and down regulation during infection between wild type and DOC5 knockout. So what's upregulated is actually suppressed because I have a lower transcription rate in the wild type compared to the knockout, so that's why it's upregulated. The other way around is when I have a smaller expression in the DOC5 knockout compared to the wild type, actually DOC5 assumes to be stimulate these particular processes. And so this is some of the idea behind our analysis to identify this particular DOC5 controlled or assumingly proposed processes by combining this network information with the knockout signatures. And so what we have here is, and that's some of the take-home message, but we will come back to this take-home message. What we think DOC5 is doing, it moderates host defense, in particular ISG, in particular this interferon-stimulated gene 20. It also modulates vesicle transport, and we find some particular genes in the neighborhood that are influenced by the knockout signatures. This is DGOLN2 and other gold genes. And then apparently also influences the mRNA processing, some of this plasmosome component. So in detail, what we have is what we actually identify and see in our analysis, and we should come to an end, is we have some of the interferon-stimulated genes are input both, of course, in the network and then in the upregulated DG, so they are all apparently suppressed. So they have a larger expression in the wild type than in the DOC5 knockout, and this is just one example of this IgG20, which actually upregulated, because the suppression can be, of course, when they are downregulated, less downregulation and more downregulation. But ISG20, it's interleukin 6 and STAT1, they're all upregulated, and you see here, this is the RNA-seq data, so from the expression data of our A549 cells. I have on the right side the wild type, and so it's less upregulated in the wild type and more upregulated in the DOC5 knockout, and this is for H1N1, the same thing goes for H3 and 2, less upregulated in the wild type, more in the knockout. Actually, so 8-fold up regulated in wild type about, and up to 20-fold up in the knockout. So apparently DOC5 suppresses in particular ISG20. This is one message what we found out. Another thing is, and this is of course speculative, but nevertheless it was rather interesting. What we also find is, for example, potential, at least what I think, feedback loop between June and DOC5, and here again we are using links information. So we see in our DOC5 network and knockout neighborhoods, again using the combined network information with the DOC5 knockout, we find June in this intersection, and what we have is it actually changes. So again it is upregulated in the wild type. So not again, it is different to the ISG, so it is actually so DOC5 actually activates June, at least in this sense. There can be other indirect measurements of and events too, and it is less expressed, or actually some are completely differently expressed in the DOC5 scenario. So there's apparently so at DOC5 some active modulation of June. On the other hand, June is a transcription factor. So we looked at the links data, check the transcription factor, but also check the response, because June is knocked out in the links data set. And what we find is it is actually upregulated in the June knockout, meaning that it is actually, so June actually apparently suppresses DOC5. But again, this is baseline, so it's taken with caution. But nevertheless, this could be some feedback loop. We haven't tested it further, and this is some what has to be tested further for further validation. But nevertheless, we find it pretty neat that there apparently is something that at least we could explain it as a feedback loop. But if it's really true, it has to be shown. And the third large process of what we think DOC5 is modulating is this vesicle process and vesicle transport. In particular, we have one vesicle-associated membrane protein 5, which is VAM5, which is actually again highly active and upregulated in the wild type, so again for H1N1 and H5N2, but not active at all. And again, we found this rather peculiar. Other genes in this neighborhood which similarly respond, but mostly they are actually down-regulated, but less so in the DOC5 knockout. So we find this trans-Golgi network protein as well as some other Golgines, and in addition we also found this clathrin code assembly complex, which is for the clathrin coded vesicle transport. And this is just a cartoon what I made how this may happen. So out from the ribosome, this should be the ribosome, the viral particles are then transported through a trans-Golgi complex using these Golgines, GOLN2, AP1S1, and then VAN5. Again, speculative, but nevertheless, it makes to some extent, at least by combining all this type of information, makes some sense. But of course, it has to be proven further. And then as the last, the last process, what we found is this mRNA processing. In this sense, we also found these lysosome members in this combined network and DOC5 knockout intersection, and not only the lysosome members but also splicing factors, and its one binding protein, one component of the exosome, and some of the nuclear pore complex. Again, it has to be tested if it's really true, but nevertheless we found it rather interesting. What we actually tested is these three key targets, ISG20, June, and VAN5, and we did it by reverse real-time qPCR. So similar to the information what we had from the RNA-seq data, we did some similar measurements and tested them by qPCR, checking in H1N1 as well as H3N2 infected cells, and comparing the response of these particular genes in the wild type and in the DOC5 knockout. And again, what you saw from the, remember from the RNA-seq data, ISG20 is active in the wild type, but much more active in the DOC5, so apparently DOC5 suppresses the expression of ISG20. The other way around is for both June and VAN5, so they are both highly active and expressed in the wild type, but less so in the DOC5 knockout. Same thing, and even more impressive, this is VAN5, so this Vesicle Transport process. So apparently this is needed and required or upregulated and modulated by DOC5, and requires actually DOC5 to be functioning, and this the expression is rather, well, it's more or less the same, and doesn't change at all in the DOC5 knockout. And this reassures us nicely what apparently was going on with DOC5 with this network analysis. And we jump over this slide. This just shows another slide of the combining the RNA, the viral proteins with our network. I just want to gloss over it and only want to say, so we combine protein-protein interaction information between the viral proteins with our constructed co-expression network, and to some extent the functions that are somehow modulated and identified in this combined network that are interacting with the particular viral protein actually makes sense of the viral function these proteins are known for. So this M1 protein, which is the early phase of viral life cycle function, are associated with this particular function in our co-expression network. Similar, this NP protein, which is the riboprotein complex, is associated with RNA transport. But I want to come to my final slide. So with all this information, we built up a cartoon of processes what apparently happen and are modulated by DOC5, and you see this cell sketch from the entry of the viral cell to the export, and so DOC5 is right here. The green nodes are in the DOC5 network, but may not have a significant overlap, or doesn't have a significant overlap with the DOC5 knockout signatures, but the blue nodes have this. So the blue nodes are all the the genes that have significant overlap between both DOC5 network as well as the DOC5 knockout signatures. And so that's what we think may happen. Again, what I showed you already before, so there's some processes for vesicle transport, in particular on the on the already translated side after mRNA processing from the nucleus, transporting the viral components out of the cell, but apparently maybe also some influence on the in the entry, but maybe not so much. But nevertheless, we find some of these targets in our network. On the other hand, we also find this interaction and pathways of and particular genes in our network which are related to the spliceosome and mRNA processing that happens in the nucleus. And so this is some of our model. To some extent we identified and validated some of the components, but of course it's more to be done. And my summary at the end of the talk is that the linked data is very useful in our analysis, but as it is based, and it has to be supplemented by targeted perturbation information that includes the additional variables, as in our case, the infectious state. On the other hand, what we found that ability DOC5 modulates these three types of cell processes, the vesicle transport, the pre-RNA processing, and apparently suppresses the host defense, but nevertheless, as I mentioned already, further analysis and further validation experiments are required. And here I just end and acknowledge, in particular, Eric Schart is our fearless leader of not anymore, but nevertheless he started a fearless leader of the ICAM Institute, and then Bing Sam, who is the PI at ICAM, but it's also a multi-location a project. So Yuri Gadin at NYU is the PI there, and then of course Wong Song, Lin-Hui, and then Cui Weng and Guy Mason and Bing Shu, who mainly did the experiments, are acknowledged, funded by NIH and NIID. This is the new Wong grant, and as I mentioned, it is now impressive its nature, a systems biology implication journal, and I thank you for your attention.
All right, great, Christian. So we have a few minutes for maybe a couple of questions. I know that Kai had a question, and I think what we were curious about, you mentioned the links data, but it was not clear to us exactly how you used it. Did you use the perturbation data? Did you use it just to create the modules? So if you can give us a little bit more technical details, and Kai, if you want to chime in and clarify a question.
So I used really the perturbation data. So I focused on the matching cell lines and used the links information and the perturbation data from the links data set for validation. And I did it in two ways. The one way is we actually started to identify what particular pathways and functions are involved, and then identified that there may be some additional targeted approaches necessary where we use our own knockout. It also helped us, which I didn't show, to identify the specific network inference approaches. So we use them as a measurement, and again we use the perturbation information from links as a measurement how valid our network inference methods are, and there again we use this intersection and intersection information of on network enrichment, network neighborhood enrichment.
This is Kai Young from University of Washington. So what perturbation data do you mean? Do you mean drug perturbation, genetic perturbation?
No, sorry, genetic perturbation. So knockout information. So knockout and overexpression, but mostly knockout, because for the overexpression we didn't have so many genes. So we only mostly used the knockout information of the matching cell lines, and it was mainly H549.
Okay, so was it L1000 data? And what level?
Did you get it from Geo and or from where did you actually?
We got it. That's a good question, because I didn't down. So we downloaded it. I'm sorry. I don't remember which particular level it was, because I didn't do the download by myself.
I see. Thank you.
So it was some time ago. So I cannot answer this question honestly.
Do you know, like, when you process the RNA-seq data, what was the pipeline that you used?
Well, the RNA-seq data, for our RNA-seq data, we used the usual so coupling stophead pipeline. But for the links data set, it was already processed. We only used the signatures. We just recalculated the DGs, but I don't remember, because I didn't do it. That's the thing.
I'm sorry. So I thought you used the links L1000 data when you compute the co-expression networks, right?
Correct. Yes.
So how could you use the signature only?
I use so the signatures are, so I combined the signatures with my own network. So I had the signatures, which genes are responding to a particular knockout, and I used the genes that are responding, so either up and down regulated of the particular knockout with the gene neighborhood of the particular knockout in my own network.
I see. So how did you compute the co-expression network? Exactly what data did you use?
The co-expression network are from the, it wasn't clear about that. The co-expression network was based on the 12 transcriptomic data sets from Geo. So from these data sets, we calculated all the 12. From the 12 data sets, we used the co-expression network, and had 12 co-expression networks, and then we used the consensus, just counting the edges.
So these 12 data sets from Geo are all H1N1, H5N1, so they are flu related.
Correct.
And so you computed the co-expression network by combining, I assume somehow you combine these 12 data sets that are flu related from Geo.
Right.
Then you use the links data, or you're looking at the differentially expressed genes from the genetic perturbation, from the knockdowns and overexpression. And you use that to overlay on top of your co-expression network?
Correct. And I used them, so that's what I tried to show in this slide. So I have my co-expression network, which is the blinking nodes here. I of course always have to match. So meaning when I use the doc5 is a bad example, because doc5 is not in the links data set. But if I have, for example, my NS1 binding protein. So I have an NS1 binding protein in my co-expression network, and then I have an NS1 binding protein which is knocked out in the links data set. And then I look at the
So did you use from the genetic perturbation data, what you're getting if you knock down gene X, you see what other genes change in response to gene X. So
correct.
Are you using that information to further refine your network? Because that also gives you the connections as well, right?
Yes, yes. In our case, we didn't not really refine the network. We tried to identify the response and of the particular target gene based on the links data. So we considered the network as given. We just were looking at the network neighborhood. If the neighborhood that is neighboring a particular target gene actually has a significance or has a significant involvement with the neighboring genes. And the significant involvement they are tested by overlap with the links data set. I hope I made that myself clear.
So my question is regarding the data integration. How did you figure out what data you want to use to compute your co-expression and then add on top? Because I would, playing the adversary here, I would argue you could also get pretty good networks by using the genetic perturbation data to start with, to compute your network.
Yes.
And maybe when you start computing the co-expression network, you could integrate the links data earlier on. So that's my question.
Yes, we could have done that too. There are only some, well, not only, we didn't choose to do so because we were focused on the infection state as additional condition. So we wanted to have a response, or at least a network, want to use an inferred network that includes the response of influenza infection. That's why we started with the inference using gene expression profiles that are actually responding, or we are calculated and recorded the response of a cell during influenza infection. But to some extent, we could have used the others, and then we just pre-selected the particular edges based on the gene expression data. So it could be both ways.
Okay, thank you.
All right.
Okay, great. Thank you so much. And I guess you can also email Christian questions if you have a pending question. It's already 10 minutes after the hour. So thank you again for participating in our webinar. Thank you again for attending. Bye.
All right. Thank you.